This method can help answer key questions in the HIV field, such as the identification of the determinants of virus transmission and pathogenesis within two of the most affected organs by the infection. In fact, the key events driving disease progression take place in tissues. The features and complexity of infection are poorly reflected by cells in circulation or isolated and infected in vitro.
The main advantage of the ex vivo tissue culture to study HIV pathogenesis is that it retains its original spatial and functional relationships between cells, although for 2 to 3 weeks.
To begin, fill a 100 mm by 20 mm Petri dish with 20 to 30 milliliters of CMT. Transfer gelatin sponges into the Petri dish using sterile forceps. Wet the sponges on both sides and allow the sponges to soak up medium for 1 minute. Press the sponges against the bottom of the Petri dish for about 2 minutes using a sterile spatula. Use sterile scissors to cut the rehydrated gelatin sponges into pieces of equal size of about 1 square centimeter. Then, use forceps to transfer 1 sponge piece into each well of a culture plate. Add 3 to 4 milliliters of CMT into each well of a 6-well plate for the tonsils, and 1 milliliter per well into a 12-well plate for the cervix. Place the plates in the incubator, set at 37 degrees Celsius, 5% CO2, and greater than or equal to 90% humidity, until the tissue explants are ready to be loaded on the sponges. Transfer the tonsils.